Good morning, dear students. Welcome to the next part of the electrophoresis technique. As we have read in the first part, it is a very useful separation technique based on the migration of the charged particle towards the opposite end of the electrode. And the rate of the migration of the particle depend on the various factors. We have also discussed the various types of the electrophoresis. The free electrolyte electrophoresis or frontal electrophoresis we have discussed. The moving boundary and the capillary electrophoresis come under this category. In this lecture, we are going to discuss now the next type of the electrophoresis, that is zone electrophoresis. In this lecture, we should cover the following topics in the zone electrophoresis, the principle of the zone electrophoresis, the different types of the zone electrophoresis, in which we should discuss the paper electrophoresis, the gel electrophoresis. The gel electrophoresis are also categorized into various types. We have to take each one type of the gel electrophoresis. That is agarose gel electrophoresis. The polyacrylamide gel electrophoresis is also there. What will be the learning outcome of this lecture? The students will be able to understand about the zone electrophoresis in the detail with its principle, types, the gel electrophoresis, including the agarose gel electrophoresis. So what is zone electrophoresis? The zone electrophoresis involves the migration of the charged particle on the supporting media. The supporting medium is used in this type of electrophoresis. As we have discussed that free electrolyte electrophoresis occurs without the supporting media, and there we allow the charged particle to move freely in the electrolytic solution. But in zone electrophoresis, the supporting medium is used to separate out the ionic molecules from the sample and it get absorbed over this medium, and it is easy to visualize the molecules which are separated. The supporting media which is used to separate out the molecules in the zone electrophoresis are paper, cellulose acetate membrane is used, the starch gel, polyacrylamide, agarose gel. So these all are the supporting media which is used in this type of electrophoresis. The components separated are distributed into discrete zones on the supporting media. The supporting media is saturated with the buffer solution. The small volume of the sample is applied as the narrow band. So we have a various bands over this supporting media by which we can easily identify the ionic molecules present in the sample. This is the most prevalent electrophoretic technique these days. In this type of the electrophoresis, the separation process is carried out on stabilizing media, that is supporting media. The various types of the zone electrophoresis is there according to the supporting media or the stabilizing media we use, that is paper, cellulose acetate, the gel electrophoresis. Even we can use capillary electrophoresis with the supporting media. The capillary electrophoresis, if we are using the supporting medium into it, it can again come up in the zone electrophoresis. And if we are using without the supporting medium, Then the capillary electrophoresis come in, moving boundary electrophoresis. So here you can able to see the mixture of the charge particle. It is the sample. It can easily understand according to the base of the size and the shape, how it is separated. The mixture of the charge molecules in the sample, it passes out, and when we apply the electric field, it separates out on the basis of the charge. Here you can able to see the positively charged molecules and the negatively charged molecules. So first, from the mixture, on applying the electric field, it separate out on the basis of the charge. So it is the charge separation, and then we have the separation on the basis of the size. So here you can easily identify the larger size to the smaller size, and we can easily go for identifying, for analyzing, even for the purification also on the basis of the charge. It can be easily separates. So this is an idea from the sample, by the method if we go for the separation, so that we can understand more easily. The gel electrophoresis is carried out in the gel casting trays, and you are able to see this type of the gel casting tray is used. It is available in a variety of the sizes, composed of the UV transparent plastic. The open ends of the trays are closed with the tape, and while the gel is being cast, then removed prior to the electrophoresis. So gel is to be fixed under this tray, which is to be carried out the migration and the movement, and after that it is removed to visualize. So the next, after the gel casting tray, there is an comb is required in the instrument, which is to be placed over the tray, and this kind of the comb is there. The comb is placed in the liquid agarose after it has been poured. Removing the comb from the hardened gel produces a series of wells used to load DNA sample. So in the tray, for loading the sample, this comb is used, and it goes for removing it when it becomes hardened, producing a series of wells. So this is the apparatus used for the paper electrophoresis, and we have the two glass plates over this filter paper is supported out, and it is immersed in the buffer solution, and these two electrodes which are connected to this battery of the 9 volts. So we have the buffer solution with the electrolyte, over which this glass plate having the mark, the pencil line from the baseline where it start the migration, and here the filter paper just absorb the molecules which goes on migration, the different speed towards the opposite electrode. And after the completion of the migration, we take out the paper, and then it is visualized. The applications of the paper electrophoresis: the serum analysis for diagnostic purpose is carried out by the paper electrophoresis. the muscle protein, myosin, the egg protein, albumin, milk protein, casein, snake, insect venoms have been satisfactorily analyzed by using paper electrophoresis. So it is specified to analyze the different kind of the proteins and the serum with the accurate results. What is the disadvantage of this paper electrophoresis? It is very time taking technique. Around 14 to 16 hours are needed for the process of the complete separation. So it is the biggest disadvantage of the paper electrophoresis. It is very time consuming method for the separation of the various molecules. The next type of the zone electrophoresis is the gel electrophoresis. It is very frequently used separation technique to separate out the charged molecules. The gel electrophoresis involve the use of gel as a supporting media for the separation of the DNA, RNA or the proteins under the influence of the electric field. It is usually performed for the analytical purposes but may be used as a preparative technique to partially purify the molecules prior to use for the other methods such as mass spectrometry, the cloning, the DNA sequencing, as well as immunoblotting. Many other analytical techniques are there. So for the purification also this method is frequently used, in which the gel as a supporting media or the stabilizing agent is used. This is most commonly used electrophoresis in the biotechnology laboratories as well as used for almost all type of the experiments in the research and the development. The gel electrophoresis is carried out in two methods. The one is the vertical gel electrophoresis, where we go for taking the vertical type of the supporting media having the electrolytic solution which is vertically arranged, and the horizontal gel electrophoresis. All the things to the electrode and the applied field and the gel, it can be used in the horizontal support. The gel electrophoresis principle, the gel means matrix which used to contain and to separate out the target molecules. In most of the cases, the gel is a cross-linked polymer on the basis of whose composition and the porosity is chosen out based on the specific weight and the composition of the target molecules. So when a potential difference is applied across the electrode of the horizontal electrophoretic tank, so as we have said that electrophoresis is carried out in two ways. Either we can take the vertical electrophoretic tank or the horizontal electrophoretic tank containing the agarose gel or the starch, or we have the polyacrylamide gel with the biomolecules such as the nucleic acids are loaded into it and the potential difference is applied. They get separated according to their molecular size. The bigger molecules have the more molecular size and the smaller molecules have the small molecular size, which move to their respective electrodes or the opposite end of the electrodes. So as the gel we go for using in the gel electrophoresis, it acts as a sieve. The sieve restrict the particles, the larger particles stay above, and the particles which are smaller to the pore size easily passes through it. Similarly, in the gel, the larger and the bulky molecules stay behind, whereas the small molecules move faster and quickly to their respective electrodes. So we can imagine this migration or the movement of the particles like a running competition or a race. The one which is thinner have a flexible body, will be at the ending point sooner, reach first than the one who is fat and the bulky. So this type of the movement and the migration can be easily imagined, and according to this movement and the migration, it allows the particles to arrange themselves in a gel. So after the completion of the migration of the particles towards the opposite end, and we take out the gel and visualize in this UV absorbance detector, and we can observe the certain bands where we can understand according to its size, the different bands we go for observing and mark it, and can find out the group or the specified molecule in the sample. So this how we have the gel visualization takes place. The gel electrophoresis can be divided into the different type according to the gel we have selected, the cross-linked polymer on the basis of the composition and the porosity, which is chosen based on the specific weight and the composition of the target molecules. That is agarose gel electrophoresis, the starch gel electrophoresis, and the polyacrylamide gel electrophoresis. So that is also called page. So according to, we can have the natural polymer also, that is starch, and which can be used in the form of gel, and the agarose, which is used to separate out the DNA molecules, and the polyacrylamide gel, that is page. Agarose gel electrophoresis use the agarose gel as a supporting medium. It is the less expensive method than the other supporting medium of the electrophoresis like cellulose acetate. We have a good separation in this technique. The agar is a complex acidic polysaccharide containing the monomers of the sulfated galactose. The agarose is a sulfate-free fraction of the agar. The gel is prepared in the buffer and spread over the microscopic slide. A small sample of the serum or the biological fluid is applied by cutting it into the gel with a sharp edge. The electrophoretic time takes about 90 minutes in the agarose gel electrophoresis. So the gel can be prepared by mixing the agarose powder with the buffer solution and heat it up to 40 degrees centigrade. It easily convert into the gel. The buffer solution also act as the electrolyte in which we just put our sample, and the gel which is also prepared in the buffer, it is spread over the microscopic slide. And the whole process require how much time? That is 90 minutes to run over this supporting media. The small size of the DNA molecules move with a low speed, and we can take the known amount of the sample, and the other which we have taken the sample are the unknown samples. The agar is a mixture of the polysaccharides extracted from the seaweeds. So agarose is a highly purified, uncharged polysaccharide derived from the agar. So agarose is prepared from the agar, which is a mixture of the polysaccharides which is extracted from the seaweeds. If we see chemically, the agarose is a disaccharide. Repeating unit is 3,6-anhydro-L-galactose. Agarose dissolves when added to the boiling liquid. It remains in the liquid state until the temperature is lowered. The pore size may be predetermined by adjusting the concentration of the agarose in the gel. So by taking the amount of the gel as a supporting media, so the pore size can be adjusted according to the concentration used in the method. The agarose gels are the fragile. They are actually the hydrocolloids, and they are held together by the formation of the weak hydrogen and the hydrophobic bonds. So they are held, their molecules, their atoms are held together by the weak hydrogen and the hydrophobic bonds. The pores of an agarose gel are large, and the agarose is used to separate out the macromolecules such as nucleic acids, large proteins, and the protein complexes. So with the size, the pore size according to which the macromolecules can be separated, because its pore size is large enough as compared to the other gel. So the macromolecules like nucleic acids, the large proteins, and the protein complexes can be easily separated out by this method. So the sample is loaded over the tray, and this how you can able to see it is used with the help of the comb, and till the sample solidify, where we have to wait for the few minutes, and then we connect all the electrode with a battery and then allowed to migrate it. So the sample is prepared and load over the tray. What are the methods involved in the agarose gel electrophoresis technique? We have to prepare the agarose gel by adding the agarose to the buffer solution, heat it at 40 degrees centigrade, then cool it, and it is easily converted into the gel. Then gel is poured into the casting tray with the comb and allowed to solidify, as you have seen in a sample preparation. Then add the running buffer into it, load the samples and mark it. Run the gel at the constant voltage until the band separation occurs. Then you can view your sample or a gel on UV light box. So we have the band observation and you can analyze the sample. So DNA is negatively charged. When placed in an electric field, DNA will migrate towards the positive pole. So as we have seen, we go for loading our sample using a comb and until it solidify in the form of well. So we have put it into the electrodes here, negatively charged and the positive. So DNA molecule is negatively charged, it's moved towards the positive end. So the agarose gel used to slow the movement of the DNA and allowed to separate by the size. So the voltage which is apply here, that is 0.126 kilovolts. So this much of the voltage is applied to separate out the DNA molecules, and the low concentration of the agarose is taken up for the better separation. And after the completion of the movement or the migration, the gel is taken out and visualized with the help of the dye. So you can see here how fast will the DNA migrate. It depends on the strength of the electrical field buffer and the density of the agarose gel. The size of the DNA, small DNA move faster than the large DNA. You can see here the large DNA which is here is kept behind, and the small DNA move faster. So according to this, how the size the DNA molecules is separate out. To visualize the agarose gel after the migration and the movement, uh, that dye is used, and the important dye, the fluorescent dye which is used to visualize the gel is ethidium bromide. So this is the ethidium bromide, the standard concentration used in staining the DNA in gels, that is of about 0.5 to 1 microgram per ml. Ethidium bromide is a fluorescent dye that intercalates between the bases of the nucleic acids and allows very convenient detection of DNA fragments in the gel. Inserting itself between the base pairs is in the double helix. To make the DNA fragments visible after the electrophoresis, the DNA must be stained with this dye. When bound to the DNA, it fluoresces under ultraviolet light, that is reddish orange color. Convenient to use this type of the dye because it can be added directly to the gel, and it is highly sensitive, detects of about 0.1 microgram of the DNA. If it is in this concentration, it can be easily detected by this dye. So here you can see the DNA ladder. It is a solution of DNA molecules are different length, and this how the bands you are able to see under the dye. The DNA ladder consists of the known DNA sizes used to determine the size of unknown DNA. So we go for using the standard size according to which we can find out the unknown sample. The DNA ladder usually contains the regularly spaced side samples, which when run on an agarose gel, it goes for appearing like a ladder. So as you have understood the agarose gel electrophoresis, let's see the advantages and the disadvantages of the agarose gel. The advantages are that gel is easily poured and does not denature the samples. Sample can also be recovered by, you can have the reuse of the sample also. The disadvantages are that the gels can melt during the electrophoresis and the buffer can become exhausted, and different forms of the genetic material may run in unpredictable forms. So at a particular time the buffer goes on for a longer period of time, the buffer can become exhausted and gel can be melt. So you have to take the measures to prevent all these disadvantage to take the advantage from the method. What are the applications of the agarose gel electrophoresis? It is used for the separation of the restriction enzyme digested DNA, including the genomic DNA, prior to the other methods. It is often used for separating RNA prior to other method other transfer. The analysis of PCR products after polymerase chain reaction, assess for the target DNA amplification, estimation of the size of the DNA molecule using as a DNA marker or a ladder which contain the DNA fragments of known sizes. As you have seen in the slide, we have a ladder. DNA fragments can be identified, allows the rough estimation of DNA quantity as well as quality. The quantity is assessed using the lambda DNA ladder, which contains the specific amount of DNA in different bands. The quality is assessed by observing the absence of streaking or the fragments. The other techniques rely on the agarose gel electrophoresis for the DNA separation, including the DNA fingerprinting. So this is all about the agarose gel electrophoresis. Then we have a starch gel electrophoresis and the polyacrylamide gel electrophoresis. So we continue with this in the next lecture. Till then, thank you and take care.